So the hemoglobin concentration for each subject was measured at the start of investigation. So we are measuring hemoglobin concentration, okay? And then at intervals of 1 week for the next 10 weeks. So we're measuring every week for 10 weeks. The first measurement was taken 2 weeks before the first EPO injection was given. Okay, very good. So this is the mean hemoglobin concentration in gram per kilogram of body mass. And then the x-axis is the time. Time here. So first EPO injection over here. So you can see that after EPO injection, there's a slight increase in the hemoglobin concentration. Okay. And then this is the final EPO injection here. So after injection, you can see that it increases. After that, it starts to drop because no more injection really. So it starts to drop. So it starts to increase for from week 2 to week 6. And then after that, after the final injection, it starts to drop already. So it's very interesting why it drops. Huh? Okay. So let's look at the question. So 4-mark question. So describe the results shown in the figure 3.1 and suggest explanation for these results. So 2 main things you need to say. Describe the result. It's just like yesterday when there are a lot of graphs being shown and you need to describe, right? Describe the graph. So in this case, the result is in the graph. Describe the result means describe the graph. Okay. And then suggest explanations for these results. So 2 parts here, describe and explain. 4 marks, that means at least 2 marks describe, 2 marks explain, right? 2 marks describe, 2 marks explain. So describe. So describe step number 1, already said step number 1 is give the general pattern. And then number 2 is quote a pair of figures to support your general pattern statement. Number 3 is probably you want to give the detail. For example, where is the peak? Where is the interception if there is anything? And then where are the gradients? So number 3 is about talking about interception, peak, gradients, and so on. So basically, I summarize it into 1, 2, 3. Then of course, when you talk about peak, interception, gradients, you have to support it with quote the figures. It can be 1 pair of figures. Well, of course, a pair of figures, yeah, correct. It can be 1 set of figures or 2 sets of figures. If you talk about the peak only 1 set of figures, right? 1 set means 1x, 1y. If you talk about the change from here to here, you have to talk about the x and y value here and x and y value here. Then you talk about the decrease, you have to talk about decrease from this x and y value to this x and y value. Okay? Am I clear? Okay, so let's talk about the general pattern. General pattern, can you all tell me what is general pattern? Tan Yun Yung?
The curve increase then decrease.
So that means general pattern is the so what is the curve? What does the curve show?
The mean hemoglobin concentration.
What about the x-axis?
From week 2 to week 7 it increases, then 7 to 10 it decreases.
So that means how are you going to put into words? I think there are 3 sections here, right? Before injection is horizontal, and after injection it starts to increase until the peak, and then after that it drops. So there are 3 sections to this curve. So how are you going to describe?
Before the injection, the mean hemoglobin concentration remains constant.
Before EPO, is it EPO? Is it called EPO? Let me see. Injection, I think it's EPO. Before EPO injection. So the mean hemoglobin remains constant, isn't it? At what value? What is the value there?
12.6
What? What?
gram per kg.
Gram per
Body
kg
mass.
what? Body mass. Yes, you have to write everything to score, including the unit there, okay? So that is before EPO. Right, so then after that, general statement again. After first EPO injection, the mean whatever increases from what value to what value? So 12.6, isn't it? 2. So what is the next value there? Thank you, Yung Yun. So plus 2 marks. So what is the next value there? Sufyan? What is the peak value here?
15 point.
15 point what?
15.2.
Okay, 15.2. So after the first EPO injection, the mean hemoglobin increases from 12.6 to 15.2. 15.2, you have to add in the unit, whatever unit, okay? So, of course, for this one, I must also include the y-axis value. So after the first EPO injection, you have to say on week number 2, right? So you can actually put after first EPO injection, you can put in bracket week 2 to denote what is the x-axis value. All right, and then you increase from this value, you're given y1 value increases to y2. You have already given your x1 value. X1 is here. So you have to give me your x2 value. Okay. This is called code of pair of figures. As you describe the general statement, you also need to give me, you need to code the specific pair of figures. That means pair of figures means x1 to x2, y1 to y2. So I got x1 here, which is week 2. And then y1, y2 is 12.6 to 15.2. So then you have to tell me at which particular week. This will give me my x2. So what is my x2 value? Week number what, Sufyan?
Week 7.
So at week 7. So in this statement, you already got describe plus code the figures. Okay. So in the same way, you can describe for number 3 lah. Number 3 is again we are talking about the general statement now. All these here are these two, because we state the pattern plus we code the figures together in the sentence. Okay, we state the pattern and we code the figures together. So it can be in the same sentence, you get 2 marks one. Okay, so number 3, I have to talk about the third part, right, where the curve decreases. So that means to say that I can say that now this one is more general already. So therefore, from week number 7 to the final week is week number 10. Okay, so then what is the pattern? The hemoglobin, so the mean, you have to copy the whole thing, mean hemoglobin concentration decreases, then from what value to what value, okay, and then plus the unit. So can you tell me decreases from what value to what value? Lim Jing-er.
Decreases from 15.2
okay, 15.2
to 13.7 gram per kg body mass.
Okay, you all must write unit. You must write unit. If no unit, you do not get the mark, okay, because it represents something here. So, and then you need to read this one very accurately, this one here. You need to read this one very accurately. So this is in the middle of the box, right? In the middle of the box, is this one correct or not? Yes, correct, 13.7, very good. Need to read accurately. So now you have already described the overall pattern already. Okay, so after that, you need to explain. You already described, so you need to explain already. Suggest the explanation why it increases and decreases. Okay, so now you know how to describe. State the pattern, quote the figures. State the pattern, quote the figures. Okay, I don't have to say the peak because just now I already said the peak value already in my description. Okay, so let's look at the explanation. Why is it that the hemoglobin concentration can change this way? So let's look at the mark scheme. So all these are descriptions. You see constant. So the constant got marked, the increase got marked, the decrease got marked. Okay, let me point out. Constant, just now we talked about the constant region, right? And then we've got an increase, then decrease description, right? So that one got give mark. Then you see the code the data, right? Code the data, right? So the data must include both the y-axis value, the y1, y2, and the x1 and x2. It's safer to quote both pairs of figures to score. Always quote both pairs of figures to score. That means x1, x2, y1, y2. Always quote both to secure the mark. Because sometimes, you see not, in this case, they put in bracket. Sometimes they don't put bracket one and all. Sometimes they don't even have got weak. Sometimes they have the weak. So to guarantee you score full marks, you have to put both y-axis and x-axis figures, the pairs there, to guarantee the mark. Because sometimes past year they give mark, they want the x-axis value. Sometimes they don't. So how do you know sometimes they give, sometimes they don't give? Give everything, okay? So now it's explanation part already. So when the EPO is being injected, which is a signal molecule, right? And the target cell is bone marrow. So it targets the bone marrow cells to increase the production of red blood cell. So EPO is a signal molecule. When it's injected, it will target the bone marrow cells to produce red blood cells. So therefore, there'll be more hemoglobin that's being synthesized. Okay? So this is to explain why the part actually increases. Now explain the part of the curve that decreases. So why does the curve decreases? So just now when they mentioned, right? Not just now, yesterday, I always said that this y-axis and x-axis is considered as a graph. So never say the graph move up and down because the graph is stagnant. It cannot move one. It's the curve that goes up and down, right? So don't say graph go up and down. Only the curve or the line inside the graph can go up and down. Please don't say graph go up and down. The line inside the graph, whether it's a straight line or a band or whatever shape, is called the curve inside the graph. It's the curve that can move up and down, not the graph. So the curve moves down. Describe, explain why it moves down. Okay, because you know that red blood cells have got short lifespan, 3 months, right? So every 3 months, they will die. So there could be a cell signaling that stops. So once you have produced enough cell, there will definitely be a signal to stop the production of the cell. So there's no more cells. So the bone marrow cells are no longer being stimulated to produce the red blood cell. So this is a process of homeostasis in our body, right? When we need more red blood cell, we will stimulate the bone marrow to produce red blood cells. When enough red blood cell is produced, there will be a negative feedback. That means it will inform our body that there's enough red blood cell produced already. It will stop the cell signaling so that it will no longer stimulate bone cells to produce more red blood cells. So that's why the number will drop. So this one has to do with, again, I always say, when you want to say the change of concentration of something, you have to look at both the input as well as the output. So why is it my red blood cell, why is it my hemoglobin concentration drop? Is because my red blood cell number drop. Why my red blood cell number drop is because I don't have input anymore. I've got output. What is the output? The output is the cell die. After 3 months, it died. Input is the bone marrow cells, the stem cells in the bone marrow. Divide and differentiate to form more red blood cell. So this is the input, this is the output. So you need to be able to see very clearly what is happening here. Then you can actually describe very clearly what is happening. Right? Both, what is the input for my hemoglobin, red blood cell? Okay, what is my input for red blood cells? Stem cells in bone marrow start to divide and differentiate. So why is it the number drop? Because there's no more input and then there's still output. So the number will drop. Okay, so with that, you need to apply this concept to many, many parts of biology. This concept is very useful using this diagram here, input and output. Okay, then increase after injection stops. What do you mean by after injection stops increases? So let's look at here. I'm not removing the line because I need the line afterwards. So just look at the graph. So after the second injection here, there is an increase in the value. After that, it stops increasing and starts to drop. So I'm supposed to describe this region. Why does it start dropping already? Why does it not increase anymore? Okay, so let's look at the mark scheme. Increase after injection stops. Hemoglobin concentration increases for 1 week after injection has finished. That means after we inject the EPO, it will take some time for the cells to be produced. So it looks like it's 1 week, right? It will be effective for 1 week. That means the bone marrow cells will be stimulated to produce red blood cell for 1 week. So that's why the red blood cell numbers will continue to increase for 1 week, even after injection. For 1 week's period, red blood cell will be produced by bone marrow. Okay, so idea of the time delay for red blood cell production to stop. That means you see after EPO is injected, this EPO molecule will take time to bind to the signal. This signal will bind to the receptor of bone marrow. It takes time for bone marrow cells to divide, correct? And then, so after cell division is cell enlargement. After cell enlargement is cell differentiation. So all this will need time before the red blood cells can be formed. And when they form the red blood cells, they will also form the hemoglobin. So from this time until this time, it takes 1 week. So that's why after injection, the red blood cell number is still increasing because it takes 1 week for the whole process to happen. That means there's a time delay for the response. What is the response? Total the concentration of hemoglobin increases is the response. Okay, time delay. So this concept is very easy for you to understand now because you've learned about the window period. Remember the window period after infection? You also look at the graph, isn't it? So there's a graph here which talk about the concentration of antibody in your blood plasma. All right. So the time period when the antigen already enter your body, but the level of antibody is 0. It only come up later. So this time after infection, right, until antibody is produced, is called the window period. So this is a window period where you cannot find any antibody, but doesn't mean there's no response in your body, right? There's still immune response in the body, but it takes time before antibody produced because so many steps involved, right? Phagocytosis, antigen presentation, clonal selection of T helper cell, then clonal expansion T helper cell, clonal expansion of selected B cells, and then plasma cells produced and antibody secreted. So many steps before antibody secreted. So once you learn about this time delay, you also know there's time delay in the rest of the processes in our homeostasis, in cell signaling until the response, until there is a response that can be detected. So one concept applied throughout becomes very easy. Okay, one concept applies throughout. Time delay for the response. AVP means any other valid point is correct. Let me erase the whole thing. So what AVP is here? Steady increase as time required for mitosis. So these are the detail, right? The response. Mitosis, differentiation, enlargement of the cell. Proliferation is enlargement of the cell or increase in the number of cells also can. Okay. Contributory factor for increase may be accumulation or increased concentration of EPO with injection. Accumulation or increased concentration of EPO with injections. What does it mean? Increase in concentration EPO for injection. That means to say time taken for after you inject the EPO into the muscles or veins until this EPO start to travel in the bloodstream, okay, and then it goes to your bone marrow. Okay, it must travel throughout the body until it goes to your bone marrow, where your stem cells are to stimulate it. Take some time. Yeah, I think that's what they're trying to say, because accumulate, you have to circulate first before it can accumulate in the bone marrow, right, which is the target cell. Increase in concentration, also same thing. You have to wait until the EPO arrive to the bone marrow, and then after that, the concentration will increase. Not because you inject, it increases. It's because it has circulated and arrived to the target region. So it increases. Right? Okay? So I think this is a very good question to learn a lot of things, right, with the graph. Learn how to describe and learn how to explain. Okay? So this is good. This looks quite easy. Shall we do this question? Yes. Fluid MoSAT model, we already said no need to do already. State the thickness of membrane. If you don't memorize, you cannot answer this question. Right? So what is the thickness of the membrane? Please type your answer. Yes, very good. I cannot see your answer yet. Hello, my handphone refused to open. Hang on. Okay, never mind. Who typed first? 7 nanometer or micrometer? 7 nanometer, my dear, not micrometer. 7 nanometer. So the first answer correct is who? Tan Yun Yun. Okay, Tan Yun Yun. Tan Yun Yun plus 2 marks. 7 nanometer. Let's recap again. 7 micrometer is the diameter of the red blood cell. Okay, 7 nanometer is smaller than micrometer, is the thickness of the membrane. Okay, so therefore you need to memorize these two because very easy. And then the size of red blood cell, sorry, the size of nucleus is bigger than red blood cell, is 10 micrometer. 10 micrometer is the size of the nucleus. Red blood cell got no nucleus, that's why it's smaller than 10 micrometer. So these are the values that you want to memorize to make it easy. Okay. Okay, so let's move on. So these are all very basic, but can you see that you really need to memorize? Because it can be asked in exam. Okay. C. List 4 features of cell surface membrane of eukaryotic cells that are not visible in figure 6.1. Do I have a figure 6.1 here? Yes, I have. All right. So what is in my figure 6.1? Let me see. What are the features that are not found? Very easy, right? Okay, I don't see a cholesterol here, so I can write there. I don't see what? Glycoprotein, glycolipid, so I can write there. Okay, so that's very easy. This is a very easy question. Okay, no need to see answer already correct. Okay. Oh, I like this one. Cell signaling again. See, one of the functions of plant hormone known as cytokinin is to act as cell signaling molecule and promote cytokinesis. Suggest how cytokinin act as a cell signaling molecule. So this one no need already. All of you know, right? The cell signaling process. Do you want to see the mark scheme or not? Any one of you? Anyone? No? No need, No need, then we skip this one because you have touched on this so many times already. Right, okay, let's move on to next. This one also very direct, isn't it? You see? Sodium ions cross cell surface membrane using facilitated diffusion or active transport. Explain why sodium ions cross cell surface membrane by this mechanism and not by simple diffusion. So you have to talk about property of this one, right? So again, properties talk about size. Talk about whether it's high, is polarity. Polarity, hydrophobic or hydrophilic. Okay? Yeah, so that will explain this answer already. Okay, very easy. Clear all. Right, next. So you got a phospholipid molecule. You are asked to do what? Explain how the structure of this molecule makes it suitable for function in cell surface membrane. You may label and annotate it as part of your answer. What question is this? This is 2016 paper 23. So let's look at mark scheme. 2016. Paper 23. Mark scheme 2000. June, is it? November, October, November. October, November, paper 23, correct? Yeah, correct, paper 23. So let's look at the mark scheme. Is this one here? Yeah, I think it's this one here. So refer to the hydrophilic polar phosphate head. So that means in your sentence, you need to say that the hydrophilic phosphate head to score the first, not only that, and hydrophobic hydrocarbon or fatty acid tail to get the first marking point. So that means to say, leche, I don't want. I just say phospholipid molecule, no mark. I just say the head, no mark. I just say the hydrophilic head, no mark. Hydrophilic phosphate head, 1 mark. Hydrophobic fatty acid tail, 1 mark. All this together, 1 mark. So it will form phospholipid bilayer. And you can talk about the fatty acid tail can be saturated or unsaturated. And then how is the phospholipid bilayer stabilized? It is stabilized by forming hydrogen bond with the water or interacting with the water. So this is the phosphate head. It will interact with the water molecule. Actually, it is the, okay, there are some carbohydrate chains which can form hydrogen bond with water because carbohydrate has got, okay, carbohydrate chains, monomers are sugar. Sugar subunits got a lot of OH molecule everywhere. So all these OH molecules can form hydrogen bond with the water. Okay, this one got OH. So they can form hydrogen bond with water so the water will be holding it throughout the entire membrane. Okay, so therefore it will give stability to the membrane, so stabilizes the membrane. So I think this part you need to pay attention, that formation of hydrogen bond by the carbohydrate chains of the glycolipid and glycoprotein will stabilize the membrane. Okay? Right. So next question. Do we still want to do question? Or already enough for you all? Okay, let me pause for a while.